to start this Solanaceae seminar online. So welcome to this Friday again. And in this opportunity, we have Nahuel Palombo. And he studied biology at the National University of Córdoba, Argentina. And since the beginning of his career, he has been interested in biodiversity and plant evolution. Currently, he is pursuing a PhD under the supervision of Dr. Carolina Carrizo Garcia and works at the Multidisciplinary Institute of Plant Biology, also in Córdoba. His research is focused on genetic diversity and evolution of the locoto Chile, Capsicum pubescens, using phylogenomic, population genomic, and cytogenetic approaches. So today he is going to present his talk about hot questions from the Andes, tracing the origin and diversification of the locoto Chile, Capsicum pubescens, of course, the Solanaceae family. So thank you, Nahuel, and now you can share your screen.
Okay, thank you for your presentation and for inviting me to participate in this seminar. And thanks to the audience for being here. And now I will share my screen. Sorry, just a minute.
Yeah, that's perfect. Oh, now.
So, sorry. My internet is not working very well. Just a minute. I will try to.
If you can just press like full mode there in the corner, I think that will work.
Yes. So, I will get off my camera. So.
Yeah, sure. That's perfect.
Perfect. Okay. Well, this talk, as Rocio has already mentioned, is entitled Hot Question from the Andes, Tracing the Origin and Diversification of the Locoto Chile, Capsicum pubescens. So in this presentation, I will share with us some comments about the long, confusing evolutionary history of Capsicum pubescens and new insights from our ongoing research. So let's go. As Gloria mentioned it in her talk some months ago, the genus Capsicum is comprised for 42 species, is a small, increasing genus from tropical and temperate areas in America, distributed for southern United States to central Argentina, and there are 5 cultivated or domesticated species: Capsicum annum, Capsicum chinense, Capsicum frutescens, Capsicum baccatum, and Capsicum pubescens. The economically most important Capsicum species are Annum chinense and Capsicum frutescens, which are nowadays cultivated in many countries around the world. Apart from that, there are other 2 species, these Capsicum baccatum and Capsicum pubescens, cultivated and consumed almost exclusively in Latin America. Furthermore, according to the current Capsicum phylogeny, this species belongs to 3 different clades: the annum clade, the baccatum clade, and the pubescent clade. And these 3 clades are collectively recognized as the crown clade of Capsicum due to they are resolved in the most terminal branches of the lineage. Well, Capsicum pubescens, this species, is the less known among the domesticated, whose origin and affinities have remained confusing along the years. Also, this Chile stands out for its morphology and growing condition, as we will see here. Well, Capsicum pubescens was described by Rulz and Pavón. Yes, and maybe yes, Capsicum pubescens was described by Rulz and Pavón at the end of the 18th century from plants located in Peru, and then the species was studied by different researchers, for example, Eschbach, Heiser and Smith, Rick, since around the middle of the 20th century. About its common name, the species is named commonly locoto or rocoto, locoto mainly in Bolivia and rocoto in Peru. This species is highly popular in Bolivia and Peru. Also, there are other common names as Chile manzano and peron in Central America. As we can see in that image, Capsicum pubescens is a large shrub plant that can grow to at least 3 meters in height, and although usually grown as an annual, it often becomes a long-lived perennial and, in this condition, may develop a woody habit. Morphologically, we have three main features or characters in the species. It is unlike other domesticated pepper, having purple to violet flowers, as we can see here, and blackish brown seeds, as we can see in this fruit. And in relation to its fruit, it produces hot fleshy berries, variable in shape, size, and color. In addition to its morphology, Capsicum pubescens is unique among the domesticated as a mid-elevated Andean species. So we can see in this photo that grows along humid Andean valleys, both east and west of the mountains of the Andes, here. We can see in that photo's environment where pubescens is cultivated. This is a photo of a harvest in Peru, a marketplace where the fruits are sold, and the added value product, this is a kind of flakes of locoto en polvo. And in the case of Central America highlands, because the species is cultivated from Northwest Argentina to Mexico, in the case of Central America highlands, it is known that the Chile was introduced at the beginning of the 20th century, and it has locally importance only since the 40s. It could be a recent introduction of the species in that territory. So in that context, we would wonder why the locoto cultivation has not spread throughout the world. Well, we can recognize two major difficulties in transferring the species. First of all, its ground requirements, because it grows in highlands with cool, free environment and long growing season. And the second reason is the fleshy nature of the fruit that leads rapid deterioration. Well, now that the species has been presented, it's possible to point out the relevance of its research as a kind of crop diversity study, and we can distinguish the relevance and several important questions. We know that it's important to improve the understanding and preservation of useful plant diversity, including genetic and phylogenetic diversity, and to do so using different approaches to characterize the germplasm is a key for the conservation and utilization of plant genetic resource, as well as for breeding programs and biotechnological approaches. In that context, the knowledge of the origin and diversification of crops is of interest from an evolutionary as well as an applied point of view, where different questions must be considered. For example, these 4 questions. Number 1, where is the most probable site or sites of domestication? Then, what wild progenitor is the most likely ancestor? 3, is the variability of the species genetically structured? And to finish, how its genetic variation distributed geographically, Well, in the case of pubescens, this species is rather special among the Chiles since it's only known as a cultigen, I mean cultivated or domesticated form, and not in the wild. The species' infrageneric placement and origin are confusing since its closest affinities are not clear. To sum up, the understanding of its evolution and its genetic diversity is still scarce. So, in that context, to address this problem with us, now I will summarize the knowledge of the affinities and evolution of Capsicum pubescens from different approaches, and then I will present some result of our ongoing research. Well, about the archaeological evidence, according to the literature, it is suggested that the 5 cultivated species were independently cultivated and domesticated as far back as 7,000 to 8,000 years before the present, either in Mesoamerica and South America. For Capsicum pubescens, this species, there are 2 oldest findings that I show you with these figures. The oldest remains are both in Peru. Moreover, pubescens was the most common chili pepper among the Indian Empire, as we can see with these sites in Peru. So, following these figures, these archaeological remains, we can say that first, further south at 3,600 meters in the Peruvian Andes lies the site of Huaynuna, the Huaynuna site in the southern Peru. Here, a light pre-ceramic house occupied beginning about 4,000 years before the present. Here we found different Chile starch grains, including Capsicum pubescens, and on the other hand, in this figure, chili pepper starches appear to be derived from pubescens, were found in Huaca Prieta site here, this site located in the desert north coast of Peru. However, given that the greatest variability of pubescens is found in Bolivian territory, it is proposed that the domestication of Capsicum pubescens took place in some region comprising southern Peru and northern Bolivia. So the domestication could have been placed in Bolivia and or Peru around 6,000 years ago. But although to date, no archaeological evidence of the species is known or reported from Bolivia. Well, according to the morphological diversity of the species, a pioneering work was the one carried out by Eschbaum in the 70s, in which he has involved in several collecting trips throughout the South American range of the species. His goal was to gain a better understanding of the variation and the evolution of Capsicum pubescens. And as a result of this expedition, Eschbaum reported that the species is highly variable throughout its cultivation range. And when we see these figures, these photographies with different kind of fruits, the first thing that comes in mind is that the species is highly variable in this characteristic of the fruits, varying both in shape and color. As for the shapes, a range of fruits from turban-shaped, for example, this fruit or this fruit here, to spherical and to elongate, for example, this fruit, exist in the species, and the fruit may have pronounced neck present or absent, pronounced neck here or an absent neck in this spherical fruit. And regarding to the color, the range in fruit color include red to yellow, red fruits, orange fruits, and yellow fruits. Moreover, analyzing the diversity in this characteristic of the fruits, size, color, and shape, Eschbach observed that the greatest degree of variation occurs in Bolivia and Peru. We can see these fruits of Colombia and Ecuador that are more homogeneous than the other ones, the Peru fruits and Bolivia fruits. And Eschbach observed the high variation in Bolivia and Peru, and the analysis of the fruit size indicates that statistically smaller fruits are found and are produced in Bolivia. So he hypothesized a place of origin in mid-elevation region in Bolivia. The smallest fruits were associated with a less domesticated form of pubescens and would be in Bolivian territory. Well, as these photos are black and white, and these express more color. And to add a little color to these bow photos, here I show you these photos taken by our team that also point out the morphological diversity of fruits in terms of shape, size, and color. And there are other characteristics of pubescence that shows some morphological diversity. We can also find some variation in the characteristic of flowers in terms of coloration of the corolla, the number of floral pieces, and also in terms of the size of the flowers. Some flowers have the purple corolla lobes, with others grade into a rose color, for example here, and still others almost lack any kind of purple pigmentation, for example this cultigen. And finally, among other characteristics that we could mention, it is possible to find some variability in the pubescence, both in stem and in the leaves, from glabrous, almost glabrous, plants to densely pubescent plants. Well, another source of evidence that also allows us to suggest an hypothetical place of origin, it is related with the wild relatives. These wild relatives grown in Bolivia and its distribution are overlapped with the range of Capsicum pubescens cultivation. On these maps, places where Capsicum pubescens is known to be grown are marked with white dots. These white dots from southern Mexico to northwest Argentina. There, in this part of the map, the range of Capsicum cardenaci, this species, is indicated in blue, and in this violet, this little violet dot, is the range of Capsicum sbauchi in the central Bolivia. And finally, in this brown area, is the range of the distribution of Capsicum eximium. So Capsicum cardenaci here around La Paz, Capsicum sbauchi in the middle of Bolivia, and Capsicum eximium is this brown area. As we can see here, part of the range of pubescens cultivation and its hypothetical center of origin in Bolivia overlap with the distribution range of the wild species that I have already mentioned, Eximium sbaugi and cardenasi. So Capsicum cardenasi and Capsicum sbaugi are endemic to these narrow areas in Bolivia, while Eximium span across a broader range here, from the yungas in Bolivia to the yungas in Argentina to the dry valleys of central Bolivia. What about the habit of these plants? In these photos, you can see the place where Capsicum eximium usually grows. For example, in this photo of the Argentinian yungas, and also in these dry valleys of central Bolivia. This photo is from Padilla in the province of Chuquisaca. And as we can see here, the species is collected, bottled, and marketed throughout southern Bolivia. And the cardinaceae fruits are also harvested and transported. These species are known by the locals as ulupicas, although they are not domesticated, they are harvested and sold, and they have economic importance. And finally, about these wild relatives, I would like to mention that Capsicum sbaugi. This species is an even more special case within this group of plants, since it has a very restricted range in central Bolivia and is little known to science to date. Carolina and collaborators recently published an article in which they provide information on its importance, its distribution, its growth habit, and also aspects related to its genetic diversity and its affinities within the group. And indeed, Capsicum sbaugi is part of the close wild relatives of Capsicum pubescens. So Capsicum sbaugi is and could be a wild ally of Capsicum pubescens. Well, as for the possibility of interbreeding that the species have, I have just mentioned that pubescens, Eximium, and cardenasi can produce hybrids. Regarding the domestication species, as is shown in this figure, the species of the genus Capsicum and its close wild relatives, based on possibility of the crossing, they have been grouped into three genetic groups or three gene pools or species complex, as can be seen in this figure. This, the Vacatum complex, the Anum complex, and again, the pubescens complex. Within the pubescens complex or pubescent gene pool, the close relationship within Eximium, cardenasi, and pubescens is supported by crossing experiments, as we can see here in these SBAU reported fruits. Well, this evidence is supported by crossing experiments which produce fertile hybrids in any of the combinations. So natural hybrids between these taxa have been reported and evaluated, and we can see these fruits described by SBAU, and at the same time, we can mention this evidence. Recent cytogenetic evidence was reported by Scaldaferro. Here, the first molecular cytogenetic evidence of hybridization between Eximium and cardenasi is provided, showing the occurrence of ribosomal genes from both karyotypes clearly distinguishable from each other in the hybrid. So it is possible to study the genome of the hybrids and the contribution of each species, Cardenaceae, and Eximium. Well, what about Esbaugi, this other plasm species? Until now, it had not been evaluated whether it was possible to carry out successful reciprocal crosses between Esbaugi and its relatives, pubescens, Eximium, and Cardenaceae. To prove this, we are currently conducting crossing experiments between this group of species using different Capsicum eximium, Capsicum cardenaceae, and Capsicum pubescens populations. And we have success. Effectively, we have found that successful reciprocal crosses are possible. The seeds obtained, for example, here are viable, and we currently have some of them in cultivation. So this result suggests that Capsicum esbaugi would have to be added to the pubescens gene pool. To this gene pool, Capsicum pubescens, Capsicum esbaugi could be placed here, can be placed here. But, and what we still don't know, or rather what we could ask ourselves, is whether these events take place in the wild. So does the same events take place in the wild? I mean, if the hybrid actually emerged naturally in the wild, or was its emergence, yes, induced only by human action? So in this sense, an interesting finding that we learned about recently consists of these plants that have a phenotype within characteristics of both Capsicum pubescens, for example, the corolla color, and Capsicum esbaugi, for example, this kind of calyx with the teeth. These small flowers and these small fruits and this calyx are similar to the Capsicum esbaugi calyx, as I just said. So plants with this morphology had already been described and suggested as a subspecies of Capsicum pubescens called Capsicum pubescens arachnoides or arachnoideum. And again, we wonder if it's possible, this possible hybrid occurs spontaneously in the nature or is due to cultivation by human. So to sum up with the wild relative and the crossing experiments, this factor led to the conclusion that the domesticated pubescens would be originated in Bolivia and that Eximium, Cardenaceae, and Espaugi is probably an ancestral gene pool, but this evidence not prove that the three taxa are the ancestors of Capsicum pubescens. So to address this question, different molecular analyses have been performed. And now, to be brief, I'm just going to focus on these two studies. In the most comprehensive phylogenetic reconstruction here on the left, this phylogenetic reconstruction of Capsicum was that we know the last and the most comprehensive hypothesis published to date. And Capsicum pubescens has resolved as monospecific lineage. Here it displays Capsicum pubescens in a strongly supported clade, as we can see here. And without any closer affinities, rendering this informal group, the purple flowered group, to be non-monophyletic. So these species, the wild relatives of pubescens, were assigned to purple corolla clade, these three species, and in the other hand, Capsicum pubescens is assigned to another clade. then no capsicum species could be considered the ancestor of the locoto Chile. Okay, and following with this study, this work deals with an analysis of diversity and genetic structure in Capsicum pubescens, in this cultivated species, in which they used microsatellite data, and they used 40 samples. The dots point to Bolivian samples, the squares Ecuador samples, and the triangles Peruvian samples. And as a result of the analysis, two clear genetic clusters were differentiated, one corresponding to the Bolivian accessions and the other to the Ecuadorian Peruvian accessions. So far, with only 40 samples, this is the most comprehensive study of pubescent genetic structure to date. In that context, we know that it is necessary to deepen the study of the origin, affinities, and genetic variability within Capsicum pubescens. Thus, our goals were to explore Capsicum pubescens origin and affinities in a phylogenetic framework, and to analyze Capsicum pubescens diversification and genetic structure. To do so, the RAD sequencing methodology was applied, generating genome-wide data from the rocoto Chile and its wild relatives or putative ancestor. Capsicum pubescens materials came from its entire range of cultivation in Latin America, from Northwest Argentina to Mexico, with emphasis on Bolivia, where the highest variability is reported. And then phylogenetic and genetic structure analysis were performed. Well, some comments about the collecting. All Capsicum pubescens materials were acquired in local markets or germplasm banks, exceptionally from some ruderal plants in Bolivia. And again, the emphasis was placed in Bolivian sources. Then new plants were obtained to apply the RAD sequencing technology. Also, we can see here the selection of the fruit and seeds and the cultivation of the plants here in Córdoba. Well, what about the results? To present these results, first I want to recover this new capsicum phylogenetic hypothesis that was already presented in Gloria and Carolina's talk last year. In this tree, Capsicum pubescens, here in purple, Capsicum pubescens, is resolved as a monophyletic group, sister to a clade, including the wild relatives, Cardenaceae, Eximium, and Spaugi, including its putative wild ancestors, I mean. And the group is named as pubescens clade now. So with new generated data, with a new sampling and thousands of SNPs, this new hypothesis and strongly supported hypothesis includes Capsicum pubescens and their wild relatives in the same lineage. Besides, this assemblage is sister to the clade that includes the other domesticated chilies and their wild relatives, all in the most derived branches of Capsicum, this branch, the most derived branch, and are collectively recognized as the crown clade of Capsicum. Well, this second phylogenetic reconstruction includes sampling of the species resolved in the crown clade. I mean, the crown clade. Here, these analyses include more samples, and the topology within the crown clade is fully congruent with that obtained using this dataset. Both reconstruction are congruent with two strongly resolved main clades, this clade and this clade. This clade includes Annum, Bacatum, and Tovari, and the other one, Capsicum pubescens, and their putative ancestor, their wild relatives. So here I will only point to the relationship within the pubescens clade. Two well-defined branches are resolved within the Locoto Chile wild relatives, Cardenaci, and the other by Eximium and Spaugi. And again, Capsicum pubescens is resolved as a strong monophyletic group. And according to the length of the branches, you can see how divergent is Capsicum pubescens lineage in relation to their wild relatives. This is really interesting, analyzing the length of the branch. Well, what about the circumscription of this group? In synthesis, according to this new hypothesis, the assembly Capsicum eximium, Spaugi, and Cardenaci comprise the closest allies of Capsicum pubescens, but that neither this or any other Capsicum species are Capsicum pubescens wild relatives. The wild ancestor of extant Capsicum pubescens may have become extinct. These are an isolated lineage. However, since the genetic diversity of a species can extend beyond its taxonomic limits, these suggestions that I have already mentioned I mean that pubescens primary gene pool includes also the wild Eximium, Spaugi, and Cardenaci, can still be supported, the circumscription of this primary or primary extended gene pool. Well, the other set of analysis, turning now to the result of the analysis of genetic structure, the genetic structure analysis within the species. On the one hand, we have a phylogenetic network built on the basis of a matrix of SNPs that includes 17 samplings of pubescens, and on the other hand, a principal component analysis with the same dataset. In the phylogenetic network, we can see that different groups are resolved within pubescens, and the groups that are resolved within the greater definition belong to the samples from Central America, and this red group from central southern Bolivia included most of the Argentinian germ plants. Also, in the Bolivian territory, here and here, it is possible to recognize a group of samples, this orange highlighted cluster. These samples corresponding to the town of Valle Serrano, located in central Bolivia between Interandean valleys, a fairly isolated with an important production of the locoto, and the other Bolivian cluster joined up samples from Coroico, Huancané, location around La Paz, including some ruderal plants. These localities are, as I just said, are the west of La Paz, that is in the central western Bolivia. These samples are located in the central western Bolivia. And to finish with this figure, we also have this group highlighted in blue that includes material from Peru and Ecuador. And finally, in the center of the network, we can see this high reticulation. We find the samples from La Paz market here. These samples have this behavior, this highly reticulated pattern. And for that reason, for that reticulated relationships cannot be assigned to any cluster. And the output of the principal component analysis is congruent with these results, showing a distribution or a grouping of samples in the space similar to that is seen in the network. Similar, we can see the Peruvian and the Central America samples here, and in the other side, the Bolivian samples. Well, by analyzing this result together, two genetic clusters could be identified. The number one, cultigens from Bolivia, and most germoplasms from Argentina, here, here, here. And the cluster number two, or the second group, cultigens from Peru, Ecuador, Central America to Mexico. And the samples of the market of La Paz are distributed across both groups, as we can see here, and within these graphics, the La Paz samples are indicated, are pointed to with these gray dots. Well, second, applying admixture models to estimate possible ancestral populations and their admixture proportion in the Capsicum pubescens individuals under this study, we obtained these results. Each graph represents the modeling according to a different K value from 2 to 5. The optimus K values, or the best model, were between a K value of 3 and 4, but each of these graphs provides interesting information. Within a value of K equal of 2, we have in red the cultigens from Bolivia and most germplasms from Argentina, and in blue, cultigens from Peru, Ecuador, Central America to Mexico again, and the samples from La Paz markets are scattered across both groups. These patterns is similar to that with the PCA and the phylogenetic network. And with higher K value, we can see, on the other hand, that the same cluster are recognized with 3, 4, and 5 K values. And well, the same cluster are recognized within the species, and on the other hand, a high proportion of genetic mixture across the individuals are present. We can see this high mixture here and here in this plot. It is also reminds to the reticulated relationships observed in the phylogenetic network. But analyzing this last graph, for example, we can recognize in light blue the samples for Central America to Mexico, in red the sample from Central Bolivia to Argentina, in orange the Valle Serrano samples, in green the cultigens from Apa, Coroico, Huancané, with some accessions from La Paz markets, and finally this blue group with high admixture. In this group we find the rest of the individual from La Paz, together with the samples from Peru and Ecuador. Then, in summary, the same genetic groups that were observed in the rest of the analysis are distinguished. And finally, I would like to return to the initial question about the possible origin and diversification in the species. To address this inquiry, up to now, we explored the phylogenetic relationship within Capsicum pubescens using a large concatenated alignment using both maximum likelihood and Bayesian approaches, and using Capsicum eximium as outgroup. In this reconstruction, several strongly resolved clades were obtained that are highlighted with these different colors by their provenance, again, being the earliest divergent one formed by rudelars from central western Bolivia, the samples from Apa. Here is Apa in that region near La Paz. All clays in this reconstruction may be segregated in groups corresponding to genetic clusters that I've just mentioned, I just showed, except from samples again, except from samples from La Paz, that are grouped into different positions here, here, or here across the tree, and depending on the settings and the amount of data, their position can vary, and also the support values can vary. So based on this result, the diversification between pubescens can also be analyzed and visualized in a geographic context in this map. Following the map, where the dot points to the sampling sites of this study, these sampling sites, it can be observed that from this region here in Bolivia, in central western Bolivia, where the samples of the early divergent clade were collected, this species has spread to the north and the south to the continent. I mean, from that region, this species started with his dispersion by the human. So the location of the samples from this early divergent clade overlaps with the range of cultivated, with the range of the wild species Cardenaci and with overlap with the range of Cardenaci and Eximium range. So this is an interesting point in terms of Capsicum pubescens and their wild relative diversity. Well, overall, from this possible center of origin or primary center of domestication of pubescens that could be placed in that region in Bolivia, the species would have been dispersed by humans to the north, this clade, and to the south, this another clade. This is a possible explanation about the origin, diversification, and dispersion of Capsicum pubescens. Yes, the dispersion in pubescens. Well, furthermore, some comments about these plants that belongs to the early diverging clade, these rhodolar plants. These plants belonging to the clade that diverged first in the phylogeny and were found growing spontaneously in the locality of Apa in the inter-Andean valleys, yes, of central western Bolivia, here in this place. Regarding their morphology, the morphology of that plants, these plants stands out because they are practically glabrous plants with small flowers, with 5 pieces, and having small fruits, as we can see here, with reduced space, reduced locules. All of these characteristics could be associated with a less domesticated form of the species, a less domesticated form of Capsicum pubescens maybe, and supports our hypothesis about the origin of these domesticated plants. So the main outcomes of this ongoing research: a new circumscription for the pubescent clade is proposed. Non-extant Capsicum species would be Capsicum pubescens ancestor, and the locoto Chile has evolved independently from its closest wild Alice species. Capsicum pubescens has a moderate genetic structure, perhaps due to low breeding management and or high gene flow between location that could be expressed the high genetic admixture that we found in the bar plots and in the phylogenetic network, for example. And it's the geographic origin of pubescent and putative center of primary domestication could be in center western Bolivia around La Paz, and the high reticulation in this area could evidence an initial territory of cultivation and dispersion towards the north and the south. Well, what are the next steps? The next steps to follow in this ongoing investigation are: explore Capsicum pubescens affinities under the coalescent model that is more appropriated in the context of interspecific and or population studies due to or because probable events of introgression or incomplete lineage sorting. At the same time, it's important to analyze genetic diversity within this recognized group or clusters to know if there are groups more diverse than others, for example. At the same time, it would be interesting to try to identify RADseq specific loci and or genomic regions subjected to selection and adaptive evolution. And we are also carrying out a molecular cytogenetic analysis within the species using new proofs to study its diversity following a different approach, a cytogenetic approach. And finally, we hope to be able to make new collections covering key areas still not sampled. For example, this area around La Paz or southern Peru. These areas are interesting. Well, finally, I would like to thank all the people who have helped me to accomplish these goals. To Gloria Barbosa for the pubescens materials and the wide relative materials that she has provided us, among other invaluable help. To Maricel Escaldaferro, also as my supervisor, for her help in the laboratory work with the cytogenetic analysis and collaborating in different ways along this time. I also have to mention collaboration with Jimena Reyes for her help in the work in Bolivian territory, and to Lisandro Fernandez for his invaluable, really invaluable help and assistance in these file explorations. And on the list, which may not be complete, I think, is the name of people who helped me here in Córdoba, Soledad, María Elena, with the maintenance of the plants, and also people from the University of Vienna Research Group who collaborated in the sequencing of the material and introducing us to bioinformatic work. And last but not least, to all the people who have donated seeds to us over the years and to the institution that provide the financial support. So thank you very much to all. And well, thank you for your attention. And now I am open to many questions, comments, whatever you want. So gracias.
Thank you very much, Nahuel. That was a great presentation. Very complete, yeah. And now we are open to questions. So if you want to, if you want me to read aloud your questions, type the questions in the chat. Otherwise, you can just type an R, and I will give you the voice to ask the question directly.
Sorry, I will try to get on my camera.
Yeah.
Here.
That's great. Yeah. Well, I do have a question. Like, you were talking about and you were talking about the different, like the wild relatives and probably like the potential ancestor of the pubescens clade. Have you done any morphological reconstruction to see how would be the fruit morphology of the ancestor of the pubescens clade?
Well, not yet. Yes. I prefer to answer in Spanish.
Maybe. Yeah, answer in Spanish, of course.
Well, yo estuve cultivando las plantas y con el trabajo de campo realizado por Carolina en diferentes mercados y diferentes regiones a lo largo de Bolivia y otros sitios, es posible reconocer un patrón a partir de ese clado que puede verse que diverge en primer lugar en la filogenia de pubescens, en esta filogenia dentro de pubescens, se pueden ver en los clados más basales esos frutos más pequeños, con flores pequeñas, con usualmente 5 piezas, o sea, un fenotipo más bien asociado a los silvestres, ¿no? A sus parientes silvestres. Y cuando nos vamos hacia ramas más que divergen a posteriori en el árbol, podemos reconocer frutos más grandes, flores más grandes, estos frutos con mayor número de lóculos, las flores con un mayor número de piezas, asociado a un síndrome de domesticación más pronunciado. Pero es una tendencia general que no ha sido probada empíricamente, digamos, con este tipo de estudios que vos mencionas.
Perdón, estaba muteada, sí. Muchas gracias, perfecto. Then we have a question of Lynn Boss. Lynn, do you want to do the question or?
Sure, I will. Wonderful presentation, great data, it's so interesting. I've been wondering for so long what the wild relatives and site of domestication of the rocoto pepper was, so thank you. My question is about Capsicum pubescens, variety Arachnoideum. And if this has been included in any of the phylogenetic or molecular genetic analyses, and if so, what did you find out?
Por el momento no, de hecho, aunque ese dato ha sido reportado años atrás por Ni, si mal no recuerdo, recientemente recibimos un correo, Gloria también lo recibió, con estas imágenes que yo mostré de esta posible subespecie de Capsicum pubescens, que morfológicamente uno podría pensar que se trata de un híbrido pubescens sbaugi, pero es un dato muy reciente y realmente esperamos poder incluirlo en alguno de nuestros estudios, ¿no? Sería muy interesante.
Sí, sí, muy interesante. ¿Y ha sido descrito esa variedad?
He visto, creo que es un trabajo que no ha sido publicado. Los datos son muy escasos, de hecho, datos sobre la corola, el color de la corola y otras características de Sbaugi han sido recientemente reportados porque es difícil encontrarla y bueno, aquí en Córdoba estamos un poco lejos para llegar a Bolivia por el momento, entonces queda trabajo por hacer y sobre también describir esta posible, no sé si subespecie, pero potencial híbrido.
Muy interesante. ¿Y viene de la región de Cochabamba?
Cerca de Cochabamba, más bien ubicado en el centro de Bolivia, en la localidad Samaipata, se llama.
Samaipata.
Samaipata, cerca de Mairana, más bien hacia Santa Cruz, creo que estaría más cerca de Santa Cruz que de Cochabamba, de Santa Cruz de la Sierra, pero sí en el centro de Bolivia, en esos valles.
Gracias.
Thank you very much, Lynn. Gracias. And then we have a question of Tecumseh. Are you there?
Yes. That was a fabulous talk. Very nice. Thank you. My question is, do you know if anybody has tried to obtain DNA from the seeds found in archaeological sites? It would be a wonderful addition to your study.
Of course. No por ahora. De hecho, dada esta amplia variación morfológica en territorio boliviano y la presencia de sus parientes silvestres, sería probable poder encontrar restos fósiles de pubescens, evidencia arqueológica de pubescens en territorio boliviano, pero todavía no ha sido reportada, y bueno, eso depende sobre las condiciones en las que se conserva el material, etcétera, pero a la fecha no. Sería interesante, ya que hay registros en Perú, poder realizar ese tipo de estudios con este ADN antiguo. Pero bueno, por el momento no tenemos acceso a esa gran valiosa información.
Gracias.
Muchas gracias. Y siguiendo esta pregunta, voy a hacer una pequeña pregunta también. ¿Qué edad aproximada tienen estos restos arqueológicos de pubescens?
y datan desde 6.500 años antes del presente a 4.000 años antes del presente, es lo más antiguo encontrado en estos dos sitios localizados en Perú, en el norte y en el sur de Perú.
Genial, muchas gracias. Luego tenemos una pregunta de Flavio Ramos. Flavio, ¿quieres hacer la pregunta o...?
Ok, buenos días, muy buena presentación.
Gracias.
Quería preguntarte qué fuente de resistencia o si tienen algo de información de la colecta que ustedes tienen. ¿Qué resistencias han mostrado?
¿A qué te refieres con
Resistencia genética. Resistencia genética. ¿Alguna enfermedad o de las colectas que tienen?
No, desde esta perspectiva más bien agronómica, podría decirlo, ya que no estoy introducido tanto en esta temática, no podría responderte esta pregunta. Lo que sí puedo decirte es que aquí en Córdoba, en el centro de Argentina, las especies son, las plantas mantenidas en cultivo son menos vigorosas, al cultivarlas tienen un comportamiento, un mayor vigor cuando son cultivadas en suelo, en condiciones abiertas, en lugar de en invernadero, pero en cuanto a la resistencia a algún patógeno, etc., no podría ampliarte nada ni responder con mayor profundidad a esa pregunta. No sé.
Ok, gracias.
No, de nada.
¿Crees que, siguiendo esta pregunta también, crees que pueden las especies silvestres tener mayor resistencia a plagas, las silvestres cercanamente relacionadas, que el mismo Capsicum pubescens o algo que puede adquirirse de estas especies silvestres?
Es posible, por ejemplo, estamos manteniendo en cultivo, a partir de un individuo de Samaipata, estamos manteniendo en cultivo, tendremos como entre 10 y 15, no recuerdo bien, individuos de Capsicum svaugi, y son muy vigorosas, tienen un crecimiento muy rápido. Habría que evaluar cómo se comportan en el invierno aquí, que hasta ahora no lo evalúo con este mayor grupo de plantas, pero lo que sí puedo decirte que son plagas que, bueno, me hacen sufrir, me hacen renegar un poco más acá, que son los pulgones, las cochinillas, estos silvestres, Eximium, Cardenasi y svaugi, no se ven tan afectados por estas plagas aquí. Y los Eximium mantenidos en cultivo acá en Córdoba, con heladas, con temperaturas bajas, han tenido una mejor resistencia que los pubescens. Eso sí, pero bueno, tenemos poquitos materiales, ¿sí? Podríamos hipotetizar que son resistentes a algunas condiciones, pero bueno, hay que seguir evaluándolo y recordar que estamos en un sitio bastante alejado de su rango nativo.
Perfecto, muchas gracias. Creo que Carolina tiene algo que decir sobre algunos trabajos de resistencia que ya existen en Eximium. Si ella está conectada, quiere decir.
Sí, no quería hablar. I didn't want to talk. No, there are some records on resistance in Eximium and I think Cardenasi that was absent in pubescens, but I don't remember the name of the author, Silvar and something. I will have to check. But maybe Nahuel will discover something when looking for adaptive signals or domestication signals in the rat loci. So maybe we discover something new. We don't know. Oh, he will discover something new.
That's great. Thank you, Carolina. And, well, that was a great presentation. I think that we don't have any more questions now. So please.
Just a last thing. I would like to congratulate Nahuel. And because he took the, how do you say, the torch, and he did the presentation all alone, and I just escaped. So congratulations.
Thank you.
Yes, thank you very much, Nahuel. That was a great presentation. Thank you, Carolina.
Sorry, because at the beginning, I was a little nervous, and my pronunciation is.
Yeah, that was great.
But I tried to do my best effort. My best.
Thank you very, very much. And yes, remember that you can reach Nahuel and Carolina through email as well, or the Google Groups, if you have any other questions. And next week, we are going to have another talk on Friday for these Solanacea seminars online. And this will be by Duban Canal from Colombia, talking about the neotropical genus Quatresia, mostly about the species diversity and taxonomic novelties. So see you everyone next week, and have a nice weekend. Bye.